Hello, we're going to talk about Chapter 1 today, the study of life. Feel free to take notes during this lecture. There will be some activities that we reach along the way, and you might want to pause the video and work them out yourself before I show you the answers. So today we're going to go over the entire first chapter. These are the objectives of Section 1 of Chapter 1. We're going to identify the shared characteristics of the natural sciences. We will compare inductive and deductive reasoning. We'll talk about the scientific method, and we'll look at the difference between basic and applied science. So the study of life, biology is a study of life, so we're going to talk about living things during this entire course. We will talk about microorganisms, the smallest things, and we'll even talk about some large organisms like ourselves, things that you can see with the naked eye. Living things came on the earth a very long time ago, but only recently have multicellular things came on earth. So the earth is green here because there are large multicellular beings like plants that live on earth and we can actually see them. So these are some microscopic organisms, and you might look at the image on the right and say, well, that's not a living thing. You're correct, but living things made those. And so what we're looking at are these blue-green algae, and they are really important when we talk about biology. A microbiologist, a marine biologist, even an ecologist, a cell biologist, bioengineer, maybe even a botanist would study these cyanobacteria. They are the first organism that put oxygen into our atmosphere, so it changed the way the Earth was for forever. And they made these, they're called stromatoliths. And so we can look back at the fossil record and see where these originated, and it was a long time ago, but they are prevalent still today. So these are, I think, in Australia, and we can go out and you can view them. And many people don't know what they are when they look at them. Okay, so biology, we already said it's the study of living organisms and it's their interactions with their environments. Science, though, can be defined as knowledge that covers general truths or the operation of general laws. So, especially when acquired and tested by the scientific method. So, what is the scientific method? The scientific method is a method of research with defined steps that include experiments and careful observations. And so we're going to get into the scientific method here in a minute, but we're going to talk about ways that we can use reasoning first. So reasoning is used in the scientific method. There are two types. So these are this is a picture of E. coli. Each of these little pill-shaped things here are single-celled organisms, so they all live independently of each other and can function independently of each other. If you're interested in E. coli or you're interested in going into maybe the medical field or agriculture science, you will probably study these, even an epidemiologist. So if you want to study things that cause us to be sick, an epidemiologist is the way to go. So you would probably look at these. So these are just some really cool pictures. All right, so natural science. Natural science are things that we can describe using theories and methods and even mathematical equations. Science that that is not a natural phenomenon, maybe it's supernatural. We aren't going to talk about those in science. So those are things that we can't explain through methods and hard evidence and mathematical equations. Those are things that we can't explain. But this down here encompasses a lot of the different realms of science. And so if we go clockwise, see if you can guess what they are. There's astronomy, there's biology, computer science, geology, logic, there's physics, chemistry, mathematics. And then, of course, there's many other fields in addition to this. My bachelor's degree is in oceanography, so I was a little bit of biology, a little bit of geology, and a little bit of physics all in one. All right, so scientific reasoning. Inductive reasoning is a form of logical thinking that uses observations to arrive at a conclusion. So it is very specific, so specific observations to a general overall conclusion. You can think of it as bottom up. Now, deductive reasoning sounds like inductive reasoning. It is a form of logical thinking that uses a general principle to forecast specific results. So we're going to have a general conclusion and then use that conclusion to apply to new situations. So it's general to specific and top down. So it's kind of the opposite. So we're going to look at some examples. One thing that I like to, that I like to try to remember these are that inductive is like adding things in to arrive at a conclusion. So inductive, we're going to add things in, add observations in. And then deductive is just the opposite. Number 5, animals deserve as humans, or animals as diverse as humans, insects, and wolves all exhibit social behavior. Therefore, social behavior must have an evolutionary advantage. So what do you think? That one's inductive. We're adding things in to arrive at a conclusion. The next one, a person notices that her houseplants are regularly exposed to music, seem to grow more quickly. As a result, she determines that plants grow when exposed. Inductive, right? Because we are arriving at a general conclusion. Humans are living and made of cells. Trees are living and made of cells. Jellyfish are living and made of cells. All newly discovered organisms that are made of cells must be living. So that one's deductive because we are making a forecast. And the next one here, it snows when there is precipitation and the air temp is at or below 32. It is currently precipitating and the temp is 35. Therefore, it is only raining, so that's deductive. And number 9 is inductive. So good job if you got those right. If those didn't seem that difficult to you, there are some harder examples in the textbook. And if you need to review those again, go back over. You can watch that video on this slide here. That might help you. Okay, the scientific method. So this is a general step-by-step process that helps us arrive at new conclusions by testing them. Okay, so the scientific method, series of well-defined steps, they're listed here. So this is an image from the textbook. Make an observation, ask a question, form a hypothesis that answers the question, and then we're going to make a prediction, and then we will test it. So we'll do an experiment. And then you'll analyze the experiment. Usually you analyze the data, you do some statistical analysis to say whether your data is good or not. And then you can support or reject your hypothesis based on your analysis. And if you don't support it, you could always try again. So there's some examples here of questions or observations. So why didn't the toaster toast my bread would be a question. Maybe you saw that the toaster did not toast your bread and it's still bread after it came out. Or do certain types of music help plants grow taller? So we're going to look at these step by step here. The first one is the toast question. So observation, the bread was toasted, or the bread was not toasted after being in the toaster, and it was turned on. So we did turn the toaster on, it ran, and it made the bing noise when it was done. So the question, why didn't my bread get toasted? So your hypothesis, my toaster didn't work because the electrical outlet is broken. So maybe something is wrong with the electrical outlet. That's what we're thinking. Okay, so we're making a prediction that we can test. Just a single prediction. The prediction, plugging in the toaster to a different outlet should fix the problem. So our test, our experiment, we're going to plug the toaster into a different outlet. Okay, so that's our test. Our analysis, we're going to analyze what happened, right? The toaster does toast the bread, so that's good. So we can conclude that our hypothesis was correct, that the outlet was in fact broken. So this is simple scientific method. Now we're going to add to this, since this is college after all, we're going to add some things in, and those are going to be different variables that are in the experiment part. So the test a hypothesis slash prediction. Um, During our experiment, we're going to have to define some variables. So the independent variable and the dependent variable are in just about every experiment, at least every experiment that you can test. So the independent variable is the variable that I test or the variable that I change. So notice that letter I, okay? So in our toaster experiment, it would be the outlet. So we had the original outlet, and then we had an outlet that that the toaster wasn't originally plugged into, right? The dependent variable results from the action of the independent variable. So I like to say the dependent variable depends upon the independent variable, okay? So it's the outcome. So what happened? What happened in your experiment? That's what it is. The toaster works or doesn't work. So in this case, our those were our two outcomes from our experiment. Either it's going to work or it's not going to work. So that's what we're trying to see. Controlled variables are things that don't change. So we are making sure that we're only testing the outlet. So we're not using other appliances. We're not testing the breaker box first, right? We're still using like the same type of bread. It might not matter, but maybe it did matter that we use the same type of bread, the same time of day, room temperature usually. Maybe the time of day and room temperature doesn't matter for this experiment, but in other experiments it would, especially if it's like plants that you're talking about or living things. The control group is a group that doesn't receive the test, so other appliances in the room maybe. I don't know. We really don't have a control group for this experiment, and sometimes you don't. The data is the graph that you produce after you run the experiment. So we always put the independent variable on the x-axis, the horizontal at the bottom. And then the dependent variable goes on the y-axis, the vertical axis. So I made a nice little bar chart here for this one. Sometimes a bar chart works for you. Sometimes it's a line graph or a scatter chart. But this one has the original outlet, right? Our first independent variable or our first experimental variable, and then our second experimental variable here. So notice that the alternative outlet, the toaster worked, right? It's marked that way. The original outlet, nope, the toaster did not work. All right, so this is an activity. If you want to pause the activity, you can go ahead and do so. You're going to put the steps of the scientific method in order. So they're about removing barnacles from boats. So we're trying to find a better way to do that. So all of these steps are steps of the scientific method for that experiment, but they aren't in the right order. So this would be the correct order up here, just for your reference. So the first step would be observation. So barnacles harm the exteriors of boats, so it would be letter A. Next, if we add anti-barnacle serum to paint, will it still keep the barnacles off? So that's C, that's our question, that's our second step. Letter F is the next. A paint made with 2% anti-barnacle serum will allow the least number of barnacles to attach to the paint, so that's our hypothesis. We can test that. It's only asking a single thing. The next one is B, so this is our experiment. And then after that, it's our analysis, right? Our data. We're going to look at what happened during our experiment. And then letter D is our conclusion. So we support our original hypothesis that 2% anti-barnacle serum works best. So notice that during our experiment, we had our control group, boat 1, so it just had regular paint. So controls are really nice to be able to compare back to see if our independent variable, our experimental variable, if it's actually working. So boat 1 and 4 had the most amount of barnacles attached. Boat 4's paint was falling off, so we had too much serum in the paint. Boat 3 had the least amount of barnacles attached. So if we had a nice little chart, we could actually count the number of barnacles on the boat and put that in the chart, and that would help show graphically and visually that the boat 3 was the best. And boat 1 probably had the most. Boat 2 wasn't mentioned here, but maybe boat 2 didn't have quite as many as boat 1 and 4. Okay, so if you want to do some more experimenting yourself, there's some practice questions here. You can name the variables and the different steps for each of these questions. But I'm not going to go over them, so we're going to move on. All right, so the process of science is peer review. So after you do an experiment, especially if you're doing undergraduate research, or maybe you're in grad school and you want to publish a paper, you can do so, and so you can publish into a peer-reviewed journal. So the process of peer review takes people who are in that field. So say you study nematodes, and it takes people from the nematode field from elsewhere, probably outside your institution, and they review your experiment. So it has to be written out. Typically they follow a format like this. And if your experiment seems sound enough to those experts in your field, then it's usually published. You can watch a video here on what peer review is if you want to see more, but you've probably heard that term peer-reviewed article a lot lately with the coronavirus and vaccines coming out. So helpful video there. It's pretty short. Okay, so our last part of 1.1 is to talk about basic and applied science. So basic science is just pure science, typically fact-based, knowledge-based, and then applied science usually uses basic to solve a problem. So problem solving is usually what applied science does. So the researchers there in the picture, they are trying to rehabilitate a monk's seal. So that's great, that's applied science, but that wouldn't have happened if we didn't have basic science, right? So we need to know like what the seal is, what eats it, what the seal eats, where it lives, if it's a social animal, if it is affected by any chemicals. Maybe we have some research already on the effects of chemicals on monk seals, if they have any medical histories, if they get different types of infections or diseases typically. So those are things that are useful to researchers, or to people who are using applied science. This man here is working on the Human Genome Project. He is using what we already know to help identify genetic diseases, and we can also look at paternity tests, so who is the father, and then crime scene analysis. So because we have the Human Genome Project, the use of genetics has gotten a lot easier, and it's more common. So we're finding more ways to use it. So that's the beauty of science, right? We find one thing, and then it makes other things easier. All right, so basic or applied science. Go ahead and pause this. I'm going to run through these really quick. Number 1 is basic. 2 is applied. 3 and 4 are basic. 5 and 6 are applied. 7 is basic, and 8 is applied. So good job if you got those correct. If you want to review those again, go ahead. Just rewind the video. Okay, so we're ending 1.1. We're going to move on to 1.2. So hopefully you've learned all of those objectives. In 1.2, we're going to talk about the properties of life. We'll talk about the levels of organization in living things. We'll look at a phylogenetic tree, and then we will look at different subdisciplines in biology. So these are the properties of life. We're going to go through these one by one. Order, responsiveness, reproduction, growth and development, regulation and homeostasis, and energy processing. Okay, so order. Order is having an organized structure. So cells are organized to tissues, tissues are organized in organs, organs are organized into organ systems, and then organ systems make an organism. Now, of course, this doesn't apply to single-celled organisms, but they still do have order in a single-celled organism. You know, they have their DNA packaged in a certain place inside. It's not just all willy-nilly floating about. So single-celled organisms still do have order, even though they are just one cell. Responsiveness to stimuli. This is a mimosa plant that you're looking at. It grows in Central and South America in the rainforest regions. And what it's showing you is its response to stimuli. Now, this is a exaggerated example, but plants do respond to stimuli. If you plant, if you have an indoor plant, you put it near the window, it's going to start to lean towards that window, right? So they're responding to the direction of the sun. So all living things will respond. There's a nice YouTube video that shows sunflowers following the sun during the day. It's really interesting. Reproduction, so living things reproduce. Asexual reproduction is very common among single-celled organisms or lower order multicellular organisms. So mitosis, budding, you can cut sweet potatoes up and make clones of those. And then, of course, sexual reproduction. We're all familiar with that. Growth and development, living things grow and they develop. So they grow from a young infant stage or young larval stage, and they grow to an adult as a result of gene instruction. The next thing, regulation and homeostasis, so we can coordinate our internal functions and regulate our internal habitat, right? And then energy process, so living things can use light energy or chemical energy to drive a metabolic process. So this little guy's munching on some corn and he's going to use the energy in the corn, the chemical energy, to drive his metabolic processes. So those are the properties of life. Pretty simple, right? Order, sensitivity, response, sensitivity or response to stimuli, reproduction, growth, development, regulation, homeostasis, energy processing. So hopefully we know a good example of each of those. Here's a review question for you. Viruses are not considered living because they blank. So what do you think? I'll give you some time to think. So viruses do not contain, or they're not cells. They're not made of cells. They also lack cell nuclei. But anyway, so they don't contain DNA or RNA. They do actually. They cannot reproduce. They can reproduce. They are not made of cells, so they need a host cell to be able to reproduce. Oops, there we go. All right, so next we'll talk about biological levels of organization, just moving on through here. First level of organization is atom, so A-T-O-M, and I don't have it on the next slide. So the first level is atom, so that's the smallest thing. So let's see, that single little white dot up here, that would be an atom. A molecule is like a water molecule, so something H2O, two small things together. So like this orange and red molecule here would be a molecule. A macromolecule is a very large unit composed of many molecules. So DNA would be a macromolecule. Next is organelle. So each of those little dots inside is the nucleus, so that's an organelle of a cell. Next is cells, then tissues. So human skin is a tissue, so this is a tissue. The purpley pink part, that's a different tissue. and then the light pink part is a different tissue. And then tissues are organized into organs. So the skin is an organ, it's the largest organ in your body. And then after organs, we have organ systems. So that could be like the immune system. Next is an organism. So a tree is an organism, you're an organism, your pet is an organism. When you have similar species or the same species together, that would make a population. So we talk about the human population because we are all the same species. We could also talk about the longleaf pine population in a forest. So we would just be talking about that one species, but multiple organisms of that one species. When we start adding in other species, that would be a community. So it would be the longleaf pines, the oak trees, the holly bushes, all of those would be a community. Ecosystems, we're going to add in the non-living things of the of the ecosystem. So that would be like the water, maybe the water temperature, the salinity of the water, the turbidity of it, can we see through it? Air temperature, weather patterns, climate, soil pH, all those things are non-living. Rocks, all of that is non-living. And then the biosphere encompasses either the entire Earth or a very large ecosystem. So like the Amazon rainforest, it could be a biosphere just because it's so large. So those are the levels of organization. So next we're going to look at an activity quick of different levels. They're not in the right order, but we're going to put them in the right order. So if you want to pause this, go ahead. We're going to go over the answers here. So the first one is hydrogen atom. The next one is water molecule. Then carbohydrate, so that is a macromolecule. Next is mitochondria, so that's an organelle. Next is a cell, so in this case an animal cell, right? If you've done the activity. Next is a connective tissue, so that is a one type of tissue. And then the liver is a tissue or an organ, not a tissue. Next is digestive system, and then elephant, organism, right? E is next, so it's a herd, so it's the same species, that's a population. Elephant herd is population. Then the African savanna is a community. And then G is planet Earth. So I think we skipped over ecosystem, but anyway, so hopefully you got the gist of that activity. All right, so moving on. We're going to finish up here talking about phylogenetic trees and different subdisciplines of biology. The phylogenetic tree that you're looking at here is a pretty basic one. It just shows the subheadings, the subcompartments of different domains. So there are 3 major domains of living things that we've divided living things into. We are in eukarya. Eukarya means true nucleus. And then there's archaea, which are pretty close to us. And then there's bacteria, which are not close to us at all. So I say close to us because of the branch points here. So bacteria, they are on their own arm of this phylogenetic tree, whereas archaeans and eukarya are on the same arm. Notice the branch point, though, is pretty far apart. So as we move up towards the top of the phylogenetic tree, that would be more present day time. Where if we go back, that would be long ago. Or so a branch point down here with archaea and eukarya, that was a long time ago. But we are still somewhat related, so we're still in the same arm. And then if we move upwards, diplomonads, microsporidia, trichomonads, those were more recent. And then flagellates and entamoeba, slime molds, ciliates, those are even more recent. And then animals, plants, and fungi kind of stem off at the same part. So we're pretty closely related, more related than you probably thought we were. Um, this black line down here, we don't quite know what this is, but it's a common ancestor. We refer to it as LUCA, last universal common ancestor. So that was the ancestor that evolved and diversified and then became bacteria, archaea, and eukarya. Notice there's no non-living things on this phylogenetic tree, so we don't put extinct things on them typically. We only put the living groups. Okay, so some questions to think about. Which domain of life are humans classified in? Well, that's our eukarya, right? Where in the phylogenetic tree would you expect to find the organism that evolved most recently? So that would be towards the top, right? So animals and plants, fungi, pretty close to the top, right? We are at the top. And then ciliates and slime molds, and entamoeba, and then flagellates, and still pretty close to the top, and then maybe some of the archaeans. The last one, provide an explanation of the straight vertical line. So last common ancestor, last unknown, or last universal common ancestor. So lastly, we're going to look at some of the subdisciplines of biology. This is a really great picture that I found online. So you can really look at, we can really look at the different subdisciplines. So this is genetics. This is like cellular. This is more organismal. Paleontology, groups, medicine. So all of these are different parts of biology. So you always think biology like, oh, they just do, like one thing, or there's just this general term biologist. And there's really not. You're usually specialized into one of these groups. So if you, like me, I studied nematodes in my graduate work, so I would be more grouped along the zoology part or even ecology. I stemmed into that a little bit. So just something to think about. Maybe you like biology and you're interested in one of these different subdisciplines, and those are something to look into for a career. I will point out that some of these are more are growing, I guess, now. So pharmacology, neuroscience, if you can see that down there, and pathology and epidemiology and biomedical research, and even these ones here, biotechnology, synthetic biology, bioengineering, immunology, all of these are very large and they are growing right now. So those would be good things to go into if you were interested in a career in biology. Other things, I've not heard of quantum biology. That's pretty new. Biochemistry is always a good one to get into. If you get a degree in biochemistry, you can pretty much do medicine or you can do research, genetic research. Those are two great fields in biology. You're always going to find somewhere to work. Genetics and bioengineering are great ways to experiment, and they're really fun. So if you want to do a lot of hands-on stuff, that would be something cool to look at. So anyway, that is chapter 1. If you didn't understand one of the objectives, I would suggest going back and reviewing the video again. But you can also read your textbook again. You can look at some videos that I post and the Quizlet and quizzes for these chapters. Okay, so that's it for chapter 1. I hope you have a great week. Make sure you do your assignments.